we are going to discuss about type 4 hypersensitivity reaction. Type 4 hypersensitivity reaction, which was described by Gell and Kump. Okay, see, in the type 1, type 2, type 3, type 4, these three are antibody mediated and this is T cell mediated. How? In the first type it is IgE mediated, in the type 2 it is IgG or IgM mediated, and in the third one it is complex mediated. Okay, see, type 4 hypersensitivity reaction is the only type of hypersensitivity reaction which does not include the antibody mediated mechanisms. That means in the fourth type there is no antibody mediation for the hypersensitivity causes. Okay, now we'll go into deeper dive, and remember, by the end of this session you will sure have a clarity about how the hypersensitivity reaction occurs. Okay, see, there is a bacteria, it entered the human body. So after it entered the human body, there are CD4 plus T cells. Okay, these CD4 plus T cells should recognize this bacteria to elect the type 4 hypersensitivity reaction. But this CD4 plus T cells can only recognize this bacteria if they are present on a special molecule known as MHC class 2, major histocompatibility complex 2. If only this bacterial antigen is present on this, the CD4 plus T cell can get activated. But where this MHC class 2 is present, it is present on macrophages or DC cells. Those are dendritic cells. Okay, now this macrophages or DC cells will present this antigen to the CD4 plus T cells. These are called antigen presenting cells. Okay, APCs, antigen presenting cells. So this antigen presenting cells also produce interleukin 12. Okay, this interleukin 12 will tell to naive T cells. Now here, what are naive T cells? Naive T cells are nothing but those are mature T cells which previously does not have any antigen exposure. These are fresh T cells. They are mature, but they did not have any previous antigen exposure. Okay, after bacteria comes and it is being presented by MHC class 2 and by APCs, the APCs also produce interleukin 12, which tells the naive T cells to transform into T helper 1 cells, and this T helper 1 cells produce memory T cells. Okay, till here the whole process is called sensitization phase. Okay, the bacteria had come into the body and then been presented by macrophage or DC cell, which are called antigen presenting cells, and APCs produce interleukin 12. Interleukin 12 tells the naive T cells to transform into T helper 1 cells, and this T helper 1 cells transform into memory T cells. Okay, then what happened? The whole phase is called sensitization phase, right? So then what happened? Whenever this bacteria comes again into the body, comes again into the body, there are memory T cells, right? This memory T cells will directly take this bacteria, that they will know that this bacteria had come into the body, and then what they do, they rapidly proliferate rapidly. Okay, and also produce chemotoxic substances. Those are tumor necrosis factor, interferon gamma and interleukin 2 and some other chemokines. Okay, now this interferon gamma, what it does is it activates macrophages. This activated macrophages will produce nitric oxide, reactive oxygen species and lysosomal enzymes. Okay, what this all do, they damage not only the bacteria but also the normal tissue. Okay, here normal tissue is damaged and also the bacteria is also damaged. Then this tumor necrosis factor, this interleukin 2 and these chemokines, they increase. See, if this is a place where the bacteria is there, in this place this tumor necrosis factor, interleukin 2 and these chemokines increase neutrophils and monocytes and causes inflammation. Okay, this tumor necrosis factor, interleukin 2 and chemokines, this will be happening. Neutrophils and monocytes will be increasing in the site of where bacteria is there, and also inflammation will be there. Okay, this whole process, where it started with CD4 plus T cells, right? Now think there is one more type also, that is CD8 plus cytotoxic T cell. This process, okay, in this process what happens, the bacteria is coming into the body. Sorry, not here. Previously we had seen bacteria, right? But now think as this antigen is viral antigen, not bacterial antigen. If it is viral antigen, this process will be started. If it is viral antigen, this CD8 plus cytotoxic T cells will get activated and take the type for hypersensitivity reaction as usual. But there is some changes here. The APCs will present this viral antigen to CD8 plus cytotoxic T cells. Then this cytotoxic plus cytotoxic T cells will produce granzymes and perforins. These two make the infected cell to land up in apoptosis. Okay, see, there is a viral antigen coming into the body, and then it is recognized by the antigen presenting cell, and it presented to the CD8 plus cytotoxic T cells, and this cytotoxic CD8 plus cytotoxic T cells had produced granzymes and perforins. This granzymes and perforins will make the infected cell to land up in apoptosis. Okay, this is the CD8 plus cytotoxic T cell mediated. But in that CD4 plus T cell mediated, what happens? There will be activation of macrophages and production of nitric oxide, reactive oxygen species and lysosomal enzymes, right? But here granzymes and perforins. Okay, there is one more also. Okay, where example we can take, it is viral hepatitis. We can take this CD8 plus cytotoxic T cell, we can take in viral hepatitis. Okay, we can take as example viral hepatitis. Okay, after this there is one more also, that is TH17. See, we saw T helper 1 cell get activated in the first process, but there is also T17, T helper cell 17. This produces interleukin 17. This interleukin 17 increase the recirculation of monocytes and neutrophils at the site of bacterial invading. So Th17 mediated one is there, and Th1 mediated Th1, which leads to macrophage activation and production of nitric oxide, ROS, reactive oxygen species, and also lysosomal enzymes. There will be formation of this. And also, what else we discussed? We discussed the CD8 plus cytotoxic T cell process, right? Okay, these three we can see in our type 4 hypersensitivity reaction. These are three different processes which can occur in this type 4. Okay, don't get confused. Okay, this is finished, and there is one more thing which is known as granuloma. Okay, I will tell clearly. See, the bacteria enter the body. After the bacteria enter the body try to destroy it. But what if it can't destroy? See, the body knows that I can't destroy the bacteria, and it also will not allow the bacteria to spread. So see, the body know that it cannot destroy the bacteria, for suppose, and if I can't kill the bacteria, I will help my body that by not allowing this bacteria to spread. If I not allow this bacteria to spread, even though I did not kill this bacteria, my body can be safe to some extent. So what this body does, see, this is the bacteria and this is in some tissue. So what happens? It activates the macrophages through type 4 hypersensitivity, and these macrophages are transformed into big epithelial like cells, not epithelial cells. It is transformed into big epithelial like cells, so they are called epithelioid cells. Okay, now here I will draw this particular diagram. See, here macrophages are transformed into big epithelial like cells. Those are known as epithelioid cells. So the first layer is made up of, why it is layering around? Because the body is doing so that the bacteria will not spread, right? So the first layer is made by modified macrophages. Those are epithelioid cells. Then the body think the one layer protection is not enough. So what it makes, this epithelioid cells there, right? These epithelioid cells fuse. Many epithelioid cells fuse and form giant cells. These giant cells, okay, see, in this epithelioid cell there is nucleus. Every epithelioid cell has nucleus. All these nucleuses will go into the periphery and form a horseshoe shaped nucleus in the giant cell. See, these are all giant cells, so they will have nucleus like this. They will have nucleus like this, like horseshoe shaped, like this. Horseshoe shaped nucleus in the giant cells. Okay, how the giant cells are formed? The giant cells are formed by the fusion of epithelioid cells and formed a layer around the bacteria. Then, okay, these are giant cells, right? These giant cells here have a specific name. Those are Langerhans giant cells. See, those are not Langerhans cells. Those are Langerhans, Lan. Okay, this Langerhans giant cells. Then surrounding it, the TH cells will be there. TH cells. Okay. Then again there will be fibrous tissue around it. Fibrous tissue. So now tell me the names. The first outermost there is fibrous tissue, and in between there are TH cells, and then there are giant cells. And then there are epithelioid cells, and there will be necrosis in the center, that is caseous necrosis in center, and then there will be bacteria. Okay, this is the structure of granuloma, and I had told what are epithelioid cells. Epithelioid cells are modified macrophages, and epithelioid cells come together and fuse, and this nucleus go to the periphery and form a horseshoe shaped nucleus. Those are Langhans giant cells, and they will form granuloma. Okay, granuloma itself is an important question. You should sure know about it. Okay, granuloma we finished. Then I will discuss about a test, that is Montauk's test. Montauk's test, it is done to know whether the patient is previously sensitized to TB or not. See, in this Montauk's test, the antigens are introduced into the skin. After 24 to 48 hours, there will be inflammation and rash here. Okay, why we are taking this into type 4 hypersensitivity reaction is our point of concern. See, in type 3 we do test for diphtheриal toxoid. Here the rash is coming because, see, we are injecting antigen, right? After this antigen is introduced into the body, this introduced antigen will combine with the previously formed antibodies. If only previously the body had fight with the diphtheриal toxoid and had antibodies only, if it had, the antibodies will immediately combine with the antigen and form the rash by forming immune complex, and it will occur in 4 to 12 hours. But here it is taking 24 to 48 hours. Why? Previously sensitization had occurred, but to elicit the response, it will take this much time, and based on time we can differentiate whether it is going to type 3 or type 4 hypersensitivity reaction. And Montauk's test, which is mediated through type 4 hypersensitivity, it is 24 to 48 hours. We can see the rash after 48 the rash we can see in 24 to 48 hours, and in type 3 we can see in 4 to 12 hours itself. Okay, this is T cell mediated rash, this is antibody antigen complex mediated rash. Okay, this is all about type 4 hypersensitivity reaction. Hope you all understood. Okay, if any problem is there, you can pin in the comment. I can repeat it once more. And okay, before pinning the comment, you have to tell which part of the concept you did not understand, so I can tell it clearly. Okay, this is type 4 hypersensitivity. Hope you are all understanding the topics. If any problem is there, you can pin it in the comment. I will react to it. Okay, thank you.